Thank you, Prof. Sanjiv. I'm taking over from Prof. Azlina. Good morning, everyone. I'm Shin Yi. To continue our session, allow me to introduce the next speaker, my colleague from the Department of Molecular Medicine, Dr. Ain Zubaidah Ayub. Dr. Ain earned her bachelor's and master's from the University of Nottingham, United Kingdom, and recently completed her PhD in molecular medicine at our very own University Malaya. Her PhD thesis focused on investigations of cellular and molecular signatures of cancer stem cells in hepatocellular carcinoma using integrative micro RNA and transcriptomics approach to identify molecular drivers and regulatory networks underlying cancer stem cell associated plasticity. Her research interest includes biology of cancer stem cells, cellular plasticity, and multi-omics approach to study mechanisms of diseases. Today, she will share with us about the significance of cancer plasticity in therapeutic resistance, metastasis, and tumor recurrence. The title of her talk is Unravelling the Molecular Regulatory Networks of Cancer Stem Cell Plasticity. Over to you, Ain.
All right. Thank you so much, Prof. Fung Shin Yi, for the kind introduction. Can everyone see my screen?
Yes, we can.
Right. Okay. Right. Assalamualaikum and good morning. I bid to everyone. Thank you so much for coming to this webinar. So my name is Ain. So today I'm just going to give a little bit of introduction on the field that I'm working on previously for my PhD, which is a cancer stem cell, and a little bit on the significance of cancer stem cell plasticity. Right, before I start, I'm just going to give like an overview or background, because I'm not sure, like I'm pretty sure like there's people of various background here. So I'm just going to talk a little bit on cancer. So as you all know, cancer is one of the leading causes of death worldwide, which accounts for 10 million deaths in 2020. And some of the type of cancers that causes death in cancer includes lung cancer, colorectal, followed by liver cancer, stomach, breast, etc. So the type of cancer that I worked on previously was hepatocellular carcinoma, which is a primary types of liver cancer. I'm just going to put some pointer option. Okay, right. And so there are multiple risk factors that are associated with increased risk of getting cancer, and that can include various exposures to physical, chemical, and biological carcinogens. And some risk factors that are important includes smoking, consumption of alcohol, exposures to UV, and chronic infections by pathogens such as viruses, bacteria, and parasites. And as we move towards aging nations, we know that increasing age can also contribute to the increasing risk of cancer, as well as there is a prevalent increase in metabolic disorder, especially in Malaysia. We have this obesity and diabetes epidemic that can also contribute to the increasing trends of cancer incidence in the coming years, right? So early detections and treatment can definitely reduce the rates of mortality in cancers, but unfortunately, some of the cancers are diagnosed at a later stage, where the cancer has already become more aggressive and has spread or metastasized to other sites of the body. So metastasis is actually the primary cause of death from cancer, and apart from that, we also have patients that actually after being treated, they can relapse, and that also contributes to the aggressiveness of the cancer itself. So one of the hypothesized contributors of the aggressive behaviors metastatic as well as therapeutic resistance in cancer is due to the presence of this population termed cancer stem cells. So I'm pretty sure some of you are familiar with the term stem cells, which are progenitor cells, which are cells that can give rise to different types of cells in our body, and you are familiar with cancer. But what are cancer stem cells? So these are actually a small subpopulation within the tumor. They can be present in a very low percentage, up to like 0.1%, and they confer stemness properties. So stemness properties refers to their sharing the stem cell properties, which is their capacity for self-renewal, which means that they have infinite capacity to proliferate, as well as the capacity to differentiate. So these cancer stem cells can differentiate into different types of cancer cells and make up the bulk of the tumor. So there has been like a lot of research that try to prove like where these cancer stem cells actually come from. So multiple hypotheses that mention that they can come from the stem cells itself, stem cells of a specific organ, or they can actually arise from progenitor cells, or they can actually come from differentiated cells through a process called de-differentiation. So they go back towards a de-differentiated cells to become cancer stem cells. Stem cells, so research in cancer stem cells has been driven by the understanding that they have stem cell properties. So a lot of cancer stem cells has been isolated based on the expression of stem cell markers in the corresponding organ. So up to now, I think there has been like dozens of cancer stem cell markers that has been used to isolate to characterize cancer stem cells in almost all types of tumor. And some of the most prominent one include the CD123, CD44, the epithelial marker EpCAMs, single site population. So the site population is the cells that are actually expressing drug transporters, so they can actually efflux the drug, and also the aldehyde dehydrogenase, which is an intracellular markers. So depending on what markers that they express, they may like exhibit like different competencies for different stem cell properties, like different aggressiveness behaviors. Right. So research in cancer stem cells has been like driven by, like the understanding of cancer stem cell biologies has been driven by a lot of research that isolate and characterize this population. So some of the main method to isolate and characterize this population has included the site population assay, which basically means that the cells are actually isolated through this method called flow cytometry, whereby only cells that are able to efflux the drug. So because cancer stem cells are resistant, they are higher, like they have higher resistance, and they can effectively more effectively efflux the drug. So this site population assay has been one of the main markers for cancer stem cells, and there's also density gradient centrifugation, and also LDH assay. But the gold standard to characterize the cancer stem cells population, usually they're isolated from clinical samples from the cancer patients, is that this population will be isolated and will be subjected in a low amount in the immunodeficiency mice. So if these cells are capable to regenerate the tumor similar to its original tumor, so these cells are considered cancer stem cells. And there are also like spheroid formation assay, which can culture the cells which are isolated either from the clinical samples or as well as those that are actually isolated from cell lines, so the cells that are established in the lab. And recently, the immunotherapy, the immunobiology of the cancer stem cells has also been like established. So one of the method has been the immune selection by natural killer cells. And in fact, there are more assays, but most of the assays are based on the isolation of the cancer stem cell markers, right? And so based on these studies, so the studies of cancer stem cells has spanned across almost 30 years now. And we have managed to identify the properties of cancer stem cells, and the major properties, of course, the one that I mentioned just now, their self-renewal and differentiation capacities. But these cancer stem cells have been characterized to be resistant based on their ability to escape apoptosis, which is a cell death program, and they are also insensitive to growth suppression. So they will still grow even without growth factors, because they are capable to produce their own growth factors to survive. And then, they also have a cell survival signal activation that regulates their uncontrolled proliferation. And usually this growth survival signal activation is due to their activating stem cell signaling pathways, which are not supposed to be activated in normal or cancer cells. When these signaling pathways are activated, because stem cells need to proliferate indefinitely, so activation of these signaling pathways in cancer cells or normal cells will cause a transformation of the normal cells into cancer stem cells. And they also have the capacity to escape immune surveillance, so they are resistant to immunity. And also there have been studies that show that cancer stem cells confer different metabolic programs, so they have a shift in their metabolism, as well as the induction of EMT. So EMT is actually a very important program, which I will talk about later. And most importantly, because of their ability to resist and proliferate indefinitely in the tumor, they are also involved in the promotion of invasion and metastasis. And cancer stem cells has also been shown to involve in the angiogenesis and lymphoangiogenesis as well, which all are the processes that are involved during the cancer progression. Okay, right. So because cancer stem cells have been shown that they drive the tumor initiation, the progression, metastasis, resistance to treatment, and eventually they will cause the tumor relapse, they are considered as the roots of cancer progression. So meaning that the targeting of this population is important in order for us to achieve a better or effective cancer treatment. So conventionally, the current cancer treatment only targets the rapidly dividing cancer cells, which cancer stem cells were capable to escape because of their quiescence properties. And when these cells are still present after the treatment, so they are able to repopulate the tumor back to cause the tumor to recur. So that's why the future cancer therapy should incorporate the targeting of these cancer stem cell populations, as well as combining with the current conventional treatment, as we have to target this root in order to ensure that the cancer that can no longer recur. So this is like the concept that they believe that targeting cancer stem cells can actually improve the treatment for cancer. Right, but it's not as easy as that. So recently, it was found that cancer stem cells itself, they are actually can be present in a different subpopulation, meaning there's a different subset of cancer stem cells that can be present within the tumor, and this can also changes across the different stages of cancer. So for the normal hierarchical model of cancer stem cell, just saying that the cancer stem cells can self-renew to produce more cancer stem cells and differentiate to produce the cancer cells, so making up the bulk of the tumor. But we now know that even cancer cells, which are differentiated, can actually gain these cancer stem cell-like properties through a de-differentiation program. And in fact, there has been some research that have shown the cells that are within the the cancer microenvironment. So we not only have cancer cells within the tumor, but we have like resident cells like fibroblast cells, endothelial cells, immune cells, etc. And these cells can actually promote the plasticity of the conversion of non-CSC to become CSC, and they themselves can actually become CSC. So this actually like generates more heterogeneity in cancer, and with this heterogeneity within the tumor, it can actually cause, it complicates the treatment for cancer, since we have multiple populations with maybe like different capacity for aggressiveness, right? So I'm just going to talk about what plasticity means. Right, so cancer plasticity means that this is the ability of the cancer cells to adopt different identities along a phenotypic spectrum. So if you're familiar with the hallmarks of cancer, it keeps like growing every year. So recently, actually last year, they have actually added 4 more new dimensions of hallmarks of cancer, and one of it is actually phenotypic plasticity. And in context of cancer stem cell plasticity, it means that it will enable the dynamic switching between CSC and non-CSC states. So this is actually one of the ways that cancer cells can adopt to survive the different stresses or pressures, because we know that during cancer progression, the cells have to grow, and it will have like requirements for a lot of things, nutrients, and there's a lot of pressures for the cells to ensure that they can continue to grow. And such plasticity will actually generate cancer heterogeneity, and this will actually cause more challenges towards treatment, as it can also contribute to treatment resistance. Right. So I'm just going to talk about epithelial mesenchymal transition. Right. So this is actually a program that has been postulated to be one of the key mechanisms by which the cells can actually confer this plasticity. So basically, epithelial mesenchymal transition is a developmental program that is actually involved in embryonic development, and in the context of cancer, this EMT is actually activated when the cells need to migrate or metastasize. So this is actually the main program whereby the cells confer the capacity to invade the surrounding tissues and migrate to other sites of the organ through the loss of epithelial features and a gain of mesenchymal features. So the cells are able to now invade surrounding tissues and then migrate towards in the circulation, either in a single cell population or either as a collective migration. And recently, the understanding of epithelial mesenchymal transition has actually evolved in a sense that the activation of EMT does not necessarily means that the cell fully lose the epithelial features and fully exhibit mesenchymal features, but they can actually be in an intermediate state whereby they can actually express epithelial and mesenchymal features at the same times. And in fact, this partial EMT has been postulated to be the more effective way for the cancer cells to migrate, because it gives them the survival capacity as they can actually migrate in a collective migration in a clusters itself. So the intermediate or partial EMT is also one of the key mechanisms that actually regulate the plasticity, as it allows the cells to be in a transient state. And this transient state can actually confer the cells with enhanced metastasis properties and also increase their therapeutic resistance. And most importantly, in the context of my study, the activation of EMT is actually one of the mechanisms by which cancer cells can also confer cancer stem cell or cancer stem cell-like phenotypes, and therefore linking the EMT and cancer stem cells plasticity. Right. So as we all know, during the cancer progression, there are different changes that happen, and the cells has to adapt to all these changes in response to multiple pressures within the tumor microenvironment. And some of the factors that actually contributes to the plasticity of the cells can be cell intrinsic factors. So the cells have to undergo like changes in their molecular at the molecular level through epigenetic modification or through transcriptional regulation or mutation. And we also have like the cell intrinsic factors. As I was saying just now, we have resident cells within the tumor microenvironment, such as fibroblasts and immune cells. And also that we have the extracellular matrix that surrounds the cell, which can actually also modulate the cancer stem cell properties. And another features of cancer is that as the cancer grows, it will become more hypoxic. And this hypoxic condition is a contributing factors for the plasticity of cancer stem cells. And not only cell intrinsic and extrinsic factors are the contributing factors, but even therapeutic pressures, such as those that come from chemotherapy and radiotherapy, can actually selectively select for cancer stem cell phenotype. So the cells that are actually survive the chemotherapy are actually those that are actually cancer stem cell-like. So with all these informations, how are we going to target this population, since we have so much factors that can actually cause the cancer cells to become cancer stem cells, and therefore will contribute to like the aggressiveness behavior of cancer. So this is the motivation behind our study, because we wanted to know like how the different pressures actually influence the plasticity features of cancer cells to confer cancer stem cell features. And in order to understand their biology, we need to know how these cells are actually being regulated, and what are the main regulatory networks that underlie this CSC plasticity. And with the fact that there's so many factors, we wanted to look at how these multiple factors, and in my project, I focus on hypoxia and therapeutic pressures, as well as the induction of EMT, looking at these different factors in how it influence the plasticity features. So this is just an overall overview of my previous study. So we use an in vitro modeling approach, whereby we establish 3 different models of cancer stem cells based on the fact that these cells can actually be enriched through different conditions. And we assess how the different modeling actually enrich the CSC properties, and how they are actually differ in terms of their CSC plasticity features. And we also use an integrated micro RNA and mRNA approach, whereby we profile these different models and look at the molecular mechanisms underlying the different CSC features in the different models. Right. So the 3 models that I worked on. So as I mentioned previously, my previous research worked on hepatocellular carcinomas. So I worked on these cancer cell lines. So we work on one cancer cell line, and from this cancer cell line, we established 3 different models. So the first model is actually a 3D model, and the second model is the EMT model, which is induced through treatment with the TGF-beta. So TGF-beta is one of the main growth factors that actually activates the EMT. And for the drug-resistant model, we use the cisplatin drug to induce the resistance, as cisplatin resistant is actually one of the main, cisplatin is actually one of the main drugs that have shown major resistance in many types of cancer. So we chose this drug. Right. So just gonna share through what my research is about. So in the first model, we try to show that how when the cells are actually cultured in 3D, which actually recapitulates the growth-related features of the cancer cells. So when we culture for 15 days, which creates a hypoxic condition, we manage to see like some markers that are actually upregulated that show that this culture actually enrich for cancer stem cells. So in this model, I use CD123, and I show that I try to optimize like the culture conditions that actually promotes the enrichment of cancer stem cells in this culture condition. I also like work on, there's another study that I did not share here, that I also look at the effect of hypoxic itself in the 3D culture, that we actually show that a prolonged hypoxic culture can actually also induce the plasticity of cancer stem cells. I'm sorry that I didn't like include that in this slide. Right. So for my second model, this is the more interesting one, because in this model, we're trying to mimic the activation of EMT in the HepG2 cells. So we found that our treatment condition can actually induce morphological, so the cellular features of the cells that are consistent with mesenchymal properties. But we do find that our treatment has were also able to retain the epithelial features. So we were able to establish a hybrid EMT features in our culture model, which actually allow us to actually interrogate the effects of the partial EMT or hybrid EMT in cancer stem cells. So we were actually quite surprised in this model itself, because initially we also thought that we need to induce like full mesenchymal, but with this model, we were able to do the hybrid EMT features. All right. So, and for the last model, we try to mimic how the cancer actually gain cancer progression, cancer plasticity through drug resistance. So we expose these cells to cisplatin drugs in 3 cycles, and we're able to get like the resistant culture. And most importantly, I just want to share like how like different culture condition actually promotes the LDH population. And we show that the CSC markers are differentially expressed in the different models. And our main like intrigue is that the EpCAM, which is actually one of the markers, are actually differentially glycosylated in the different models. That means that potentially the different pressures can also affect the abnormal glycosylation of cancer stem cells. And another important thing is that we show that the increased stemness, invasion, migration, and chemoresistant as the key processes that are common between these 3 models, whereby the EMT and proliferation features are differential. Right. So this is just to show like the approach that we take, whereby we talk about the micro RNA mRNA integration to look at the molecular regulatory network of cancer stem cells. So just gonna introduce a little bit on why we choose microRNA. So this is actually an endogenous or short non-coding RNA that can actually regulate gene expression by either targeting the mRNA degradation or inhibit the translation. And microRNA has been shown to be highly deregulated in cancers. And the interesting thing about microRNA is that because of their ability to bind to the mRNA in a non-complementary manner, it can target like many targets or many mRNA. So it can regulate many genes, and its role in cancer progression has been evidenced in a lot of processes. So microRNA itself has a clinical significance in the fact that they has been used as a biomarkers, especially circulating microRNAs, and some of the microRNA signatures has been used for diagnostic and prognostic tools, as well as some therapeutics has been developed based on the principles of microRNA being an antagonist, and some of these has been been into clinical trials, right? So So we show that the the CRC models are actually molecularly distinct between the different models as well as with the non-CSC. And I just want to like go through like the key findings from our study previously is that we were able to identify like key microRNAs in the different models. And what we found very interesting is that despite their differential the differential microRNAs being found in the different models, we found a shared pathways which are involved in the regulation of cancer stem cells. So we use a network analysis, which I will not detail in more detail here. So essentially, the study we were able to like, identify like several microRNAs and hub genes that we find as a critical regulators in CIC models, and we believe that these regulators will play a critical roles in CIC plasticity and regulation of networks associated with biomechanical properties and cell cycle machinery of cancer cells. So these are some of the networks that we are interested to look into the mechanism going forward. And yeah, so with that, this is just to offer like some perspective on how we can actually use in vitro modeling to study the biology of cancer stem cells, and it can actually have a potential to be used as a tools for drug discovery, and that we can understand a better biology of cancer stem cells through the identified regulators. And because this studies is more in the in vitro stage, we'd love to also like see how these cancer stem cells can actually be brought, the research of cancer stem cells can be brought forward with more involvement in the clinical part, especially when we look about, when we know that now that there has been expensive data that we can actually utilize, maybe using bioinformatic tools to actually study the disease mechanism, so that eventually we can use to develop therapeutics and treatment strategy for effective cancer treatments. So with that, I'm just think I've run out of time, so I'm just gonna just putting this here as because I'm actually just starting out, and these are some of my research interests. And the ongoing studies in our lab is we're still looking at the hypoxic 3D culture, and going forward, I would love to investigate the molecular mechanisms of the partial EMT, cancer stem cells, as well as therapeutic resistance by looking at the different regulators and using the CIC-based models for drug discovery, as well as through leveraging the bioinformatic analysis, so that we can actually use the clinical database and to use that as a strategy to study disease mechanism as well. Right. With that, that's all for my talk. I hope I have given maybe some idea of what cancer stem cell is and how we can actually use this knowledge to drive our understanding of how cancer actually progress and metastasize. So thank you so much for your attention.
Thank you, Ain, for the informative talk and for sharing with us on how we can actually leverage preclinical models and integrative omics approaches to understand the molecular basis behind CSC plasticity. For the benefit of our audience here today, our department is active in transdisciplinary research, and we welcome collaborations. So please do contact Dr. Ain and her team if this field of research is of your interest. We now take a few questions. We have one question from Prof. Tunku Sara. She asks, what is the relationship of single meds coming out years after remission with CSC?
Yes, that's a very interesting question. So I believe Prof is talking about single metastasis, right, years after remission. So one of the features of cancer stem cells that has yet to, we still don't understand how they can actually confer the quiescence resistance. So years after, when the cancer after treatment, some cells can actually become dormant, and also like resistance. So there has been like some research that have shown that some CSCs can actually confer this dormancy, and in response to different signals, they can actually reactivate and regrow the cancer back. So in fact, yes, cancer stem cells dormancy is actually an area of active research where they try to understand how actually after years of remission, this cancer can actually grow back.
Okay, thank you, Ain. I hope that helps. There's another question from Dr. Ang. How do you connect these miRNA data to the histone regulatory event during cancer stem cell development?
Histone regulatory event. I'm not very sure about the histone regulatory event, but in fact, when we talk about histone, that's on the epigenetic regulations. That is an area that I'm not very like sure of, but I think the micro RNA does have some epigenetic functions, but in detail, I'm not very sure about how it regulates the histone modification.
Okay, I don't see any other questions in the Q&A nor the webinar chat. So thank you so much, Ain. Thank you everyone for joining us today. If you have any questions, please do contact Dr. Ain from the Department of Molecular Medicine in our faculty. Thank you so much.
Thank you everyone.